Good morning, students. Let us continue with the chapter number 13. Next topic, Kelvin cycle, C3 cycle. Kelvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was generated. Let us now see how the Kelvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that the Kelvin pathway occurs in all photosynthetic plants. It doesn't matter whether they have C3 or C4 pathways. For ease of understanding, Kelvin cycle can be described under three stages: carboxylation, reduction, and regeneration. First step is carboxylation. In this step, fixation of carbon dioxide into a stable organic intermediate is done. Carboxylation is the most crucial step of the Kelvin cycle, where carbon dioxide is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of two molecules of 3-phosphoglyceric acid PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase, or in short form it is known as Rubisco. It is one of the most abundant enzyme in the biological world. Second step is reduction. There are a series of reactions that lead to formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction per carbon dioxide molecule. The fixation of 6 molecules of carbon dioxide and 6 turns of the cycle are required for the formation of 1 molecule of glucose from this pathway. Regeneration, the last step in which the carbon dioxide acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. So it is recycled. For uninterrupted cycles, the regeneration steps require 1 ATP for phosphorylation to regenerate the RUBP. So in all, 3 molecules of ATP and 2 molecules of NADPH are required per cycle to fix 1 molecule of carbon dioxide. Here is the cyclic representation. RUBP, ribulose biphosphate, receives carbon dioxide and water, which carboxylation occurs and forms 3-PGA, after which reduction occurs and ATP plus NADPH are utilized. Triose phosphate is formed, which gives starch glucose, and afterwards the RUBP is regenerated. Hence, for every carbon dioxide molecule entering the Kelvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of cycle are required. How many ATP and NADPH molecules will be required to make 1 molecule of glucose? So we will multiply each value of ATP and NADPH by 6. So 6 3s are 18. 18 molecules of ATP and 6 2s are 12. So 12 molecules of NADPH will be required to make glucose through Kelvin pathway. It might help you understand all of this if we look at what goes in and what comes out. So goes in 6 molecules of carbon dioxide, 18 ATP and 12 NADPH. What comes out is 1 molecule of glucose, 18 ADP and 12 NADP. Here is the C4 pathway. Plants that are adapted to dry tropical region have the C4 pathway mentioned earlier. Though these plants have C4 oxaloacetic acid as the first carbon dioxide fixation product, they use the C3 pathway or the Kelvin cycle as the main biosynthetic pathway. Then, in what way are they different from C3 plants? This is the question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They can tolerate higher temperatures. They show a response To highlight intensity, they lack a process called photorespiration and have greater productivity of biomass. Let us understand these one by one. Study vertical sections of leaves, one of the C3 plant and other of C4 plant. We will find some differences. The particularly large cells around the vascular bundles of C4 plants are called bundle sheath cells. The leaves which have such anatomy are said to have Krenz anatomy. Krenz means wreath and is a reflection of the arrangement of cells. The bundle sheath cells may form several layers around the vascular bundle. They are characterized by having a large number of chloroplasts, thick walls, impervious to gaseous exchange, and no intercellular spaces. You may like to cut a section of the leaves of C4 plants, maize, or sorghum to observe the Krenz anatomy and distribution of the mesophyll cells. It would be interesting for you to collect the leaves of diverse species of plants around you and cut vertical sections of them. Now study the pathway shown in the figure. The pathway has been named as Hechen's-like pathway. Again, a cyclic process. Let us study the pathway by listing the steps. First, the primary carbon dioxide acceptor is a 3-carbon molecule phosphoenol pyruvate and is present in the mesophyll cell. The enzyme responsible is PEP carboxylase or pepcase. It is important to register that mesophyll cells lack rubisco and the first compound formed is oxaloacetic acid. It is a 4-carbon compound like malic acid or aspartic acid in the mesophyll cell itself, which are transported to the bundle sheath cells. In the bundle sheath cell, these C4 acids are broken to release carbon dioxide and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophyll where it is converted to PEP again, thus completing the cycle. The carbon dioxide released in the bundle sheath cells enters the C3 or Kelvin pathway, a pathway common to all plants. The bundle sheath cells are rich in an enzyme ribulose biphosphate carboxylase or oxygenase rubisco, but they lack pepcase. Thus, the basic pathway that results in the formation of sugar, the Kelvin pathway, is common to C3 and C4 plants. Did you note that Kelvin pathway occurs in all the mesophyll cells of the C3 plants, and in the C4 plants it does not take place in the mesophyll cells, but it does so only in the bundle sheath cells. Let us have a look on the cycle. Here are the two cells. The upper one is the mesophyll cell which lacks rubisco, but the lower one is bundle sheath cell which consists of rubisco. In the mesophyll cell, when the carbon dioxide is accepted, it is fixed into a C4 acid. This C4 acid is transported to the bundle sheath cell where C3 cycle occurs in the presence of rubisco, and then the C3 acid is returned back to mesophyll cells. Next topic is photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plants, that is photorespiration. To understand photorespiration, we have to know a little bit more about the first step of Kelvin pathway. The first carbon dioxide fixation step. This is the reaction where RUBP combines with carbon dioxide to form 2 molecules of 3-PGA. Here we can see the reaction. RUBP plus carbon dioxide in the presence of rubisco gives 2 molecules of 3-phosphoglyceric acid. Rubisco, that is the most abundant enzyme in the world, is characterized by the fact that its active site can bind to both carbon dioxide and oxygen. Hence the name rubisco carboxylase and oxygenase. Rubisco has much greater affinity for CO2 when the CO2 is to O2 is nearly equal. Imagine what would happen if there is not so. There will be a competition between carbon dioxide and oxygen. In C3 plants, some oxygen does not bind to rubisco and hence CO2 fixation is decreased. Here, RUBP, instead of being converted to 2 molecules of PGA, binds with O2 to form 1 molecule of phosphoglycerate and phosphoglycolate in the pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugar nor ATP. Rather, it results into the release of carbon dioxide with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known yet. In C4 plants, photorespiration does not occur because they have different mechanisms. Increase of CO2 occurs in mesophyll, while fixation of CO2 occurs in bundle sheath cells. So, there is a difference in the location and hence there are no chances of photorespiration. Now that you know that C4 plants lack photorespiration, so you probably can understand why productivity and yield are better in C4 plants. Based on the discussion, we should differentiate C3 and C4 pathway. There are some factors that affect photosynthesis, and understanding of these factors that affect photosynthesis is necessary. Rate of photosynthesis is very important, determining the yield of the crop plants. Photosynthesis is under the influence of several factors. Factors may be internal or external. Internal that are in the plant may include number, size, age, orientation of leaves, mesophyll cells, chloroplast, internal CO2 concentration, and amount of chlorophyll. The plant or internal factors are dependent on the genetic predisposition and growth of plants. The external factors include availability of sunlight, temperature, CO2 concentration, and water. As the plant photosynthesizes, all these factors will simultaneously affect its rate. Hence, though several factors interact and simultaneously affect photosynthesis or carbon dioxide fixation, usually one factor is major cause or is the one that limits the rate. Hence, at any point, the rate will be determined by the factor available at suboptimal levels. When several factors affect any biochemical process, Blackman's law of limiting factor comes into effect. That is कि जो भी सबस्ट्रेट या जो भी रिएक्टेंट लिमिटेड अमाउंट में होता है, रेट ऑफ रिएक्शन उसी पर डिपेंड करता है. जो चीज़ अबंडेंस में है उस पर कभी भी रेट डिपेंड नहीं करता. सी, it states that if a chemical process is affected by more than one factor, then its rate will be affected by the one which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite the presence of green leaf and optimal light, CO2 conditions, the plant may not photosynthesize. If the temperature is very low, this leaf, if given the optimal temperature, will start photosynthesizing. First factor is light. We need to distinguish between the light quality, light intensity, and the duration of exposure to light. While discussing light as a factor that affects photosynthesis, there is a linear relationship between incident light and carbon dioxide fixation rates at low intensities. At higher light intensities, gradually the rate does not show further increase as other factors become limiting. What is the interesting to note is that light saturation occurs at 10% of the full sunlight. Hence, except for the plants in shade or in dense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of chlorophyll, that is photooxidation of chlorophyll, and a decrease in photosynthesis. Here is a graph representing the light intensity and rate of photosynthesis. We can see as the light intensity increases, rate of photosynthesis increases, but at a maximum value, it becomes stable, no increase in rate. Next factor is carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of carbon dioxide is very low in the atmosphere, that is between 0.03 to 0.04%. Increase in concentration up to 0.05% can cause an increase in carbon dioxide fixation rates. Beyond these levels can become damaging over longer periods. The C3 and C4 plants respond differently to CO2 concentrations. At low light condition, neither group responds to high CO2 conditions. At high light intensity, both show increase in rate of photosynthesis. What is important to notice, C4 shows saturation at about 360 microliter per liter, while C3 responds to increased concentration and saturation is seen only beyond 450 microliter per liter. Thus, current availability of carbon dioxide becomes limiting factor for C3 plants. The fact that C3 plants respond to higher CO2 concentration by showing increased rate of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yields. Next is temperature. The dark reaction being enzymatic are temperature controlled, though the light reaction are also temperature sensitive. They are affected to a much lesser extent. The C4 plants respond higher temperatures and show higher rate of photosynthesis, while C3 plants have a much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depend on the habitat that they are adapted to. Tropical plants have higher temperature optimum than the plants adapted to temperate climates. Next factor is water. Even though water is one of the reactant in the light reaction, the effect of water as a factor is more through its effect on plant rather than directly on photosynthesis. Water stress causes the stomata to close, hence reducing CO2 availability. Besides, water stress also makes the leaves wilt, thus reducing the surface area of leaves and their metabolic activity. This was all about photosynthesis.